Welcome to this explainer. Today, we're taking a literal journey inside the human body to uncover the sheer mechanical genius of how we move. We're going to break down exactly how millions of microscopic engines coordinate in mere milliseconds, just to help you lift an arm, take a single step, or propel yourself through the world. We'll unpack the complex machinery of your muscular and skeletal systems piece by piece, so you can really grasp the incredible biology of human locomotion. Okay, let's kick things off with a really crucial distinction. In biology, simple movements happen all the time, right? Like a single-celled amoeba shifting its protoplasm, or, your heart continuously beating in your chest. Locomotion, on the other hand, is a highly specific voluntary action that we use to actually change our physical location. Think walking, running, or climbing. So the biological rule of thumb here is that all locomotion is movement, but absolutely not all movement is locomotion. To pull off true locomotion, we need the perfectly synchronized activity of our neural, skeletal, and muscular systems. And the driving force behind this locomotion? Well, it relies on three totally distinct types of muscle. First, you've got your skeletal muscles. Those are the striped ones under your conscious control that primarily drive our posture and locomotion. Now, that's vastly different from the involuntary, smooth, visceral muscles lining your digestive and reproductive tracts. Those guys quietly push food and cells along without you even having to think about it. And finally, you have your cardiac muscles, uniquely branching, striated, and tirelessly pumping your heart without any direct conscious neural control. Now if we zoom way down into a single skeletal muscle bundle, we find these fascinating parallel filaments called myofibrils. They have this distinctly striped, alternating pattern of dark and light bands. But that pattern isn't just for show. It's the mechanical result of how two super important proteins are distributed. You have actin, which makes up the lighter I bands, and myosin, forming the darker A bands. Those light actin bands? They're actually bisected by a remarkably elastic fiber called the Z-line. And the microscopic space right between two successive Z-lines is known as a sarcomere. This right here is the absolute core of our explainer today. The sarcomere is the actual functional unit of contraction. It's the microscopic engine where the true magic of human movement actually starts. Inside that tiny sarcomere, let's look closer at the thicker myosin filaments. Because they aren't just stiff, passive rods. No way. They are complex, polymerized proteins equipped with globular heads that project outward at regular intervals, which we call crossarms. Think of these tiny globular heads as active ATPase enzymes. They're perfectly designed with two crucial features: specific binding sites to grab hold of ATP energy, and active sites to firmly latch onto those thinner actin filaments we talked about. So how does all this microscopic machinery actually create a flex? It operates through what's known as the sliding filament theory. Basically, in a fraction of a second, a signal from your central nervous system triggers a rush of calcium ions. This calcium binds to a protein on the actin called troponin, which unmasks its active sites. Boom, the energized myosin heads instantly grab those exposed sites, forming a cross bridge. Then, the myosin heads rotate, forcefully pulling the actin filaments inward. Finally, a new ATP molecule binds to the myosin, breaking the bridge so the whole cycle can reset. It is a rapid-fire cellular dance happening millions of times every single second. And this perfectly illustrates the absolute mechanical genius at play here. Because the myosin heads are simply pulling the actin inward toward the center, the actual length of the thick myosin rods, that dark A band, never changes size. Instead, the lighter I bands shrink as the Z lines are pulled closer together. Think of it exactly like rowers in a boat. The oars, the myosin, don't shrink, they simply grab and slide the water, the actin, past them to create movement. Of course, to fuel all that continuous, energetic rowing, your body needs a massive amount of ATP. And your muscles handle this demand using two distinct types of fibers. On one side, we have red fibers. These are loaded with mitochondria and a red oxygen-storing pigment called myoglobin, which makes them absolutely perfect for sustained aerobic endurance, like running a marathon. On the flip side, we have paler white fibers. These have way less myoglobin and fewer mitochondria, so they rely on anaerobic energy when oxygen is scarce. That's what powers quick, explosive bursts of movement, like a sprinter bolting off the starting block. But here's the thing, millions of microscopic sliding engines are basically useless without a solid chassis to pull against, right? Zooming back out to the macro level, those muscles eventually anchor to a massive, highly organized framework, your skeletal system. We're talking about a matrix of specialized connective tissue hardened by calcium salts, consisting of exactly 206 bones in the adult human body, plus a few specialized cartilages. This complex 206-bone architecture is elegantly divided into two principal systems. First is the central axial skeleton. That's 80 bones along the main axis of your body, forming your core, your skull, the protective vertebral column for your spinal cord, the sternum, and the ribs. Second is the appendicular skeleton. This includes your limb bones, your arms and legs, and the pectoral and pelvic girdles that attach them to the axial core. These act as the vital levers for literally all of your locomotion. Think about the architecture of the rib cage for a second. It's such a fascinating example within the axial skeleton. You have 12 pairs of ribs, but they aren't all attached equally. The first 7 pairs are true ribs, anchoring directly to the sternum with hyaline cartilage. Then the 8th, 9th, and 10th pairs are false ribs. They don't reach the sternum directly. Instead, they share cartilage to attach to the 7th rib. And incredibly, the final 2 pairs, the 11th and 12th, have no ventral attachment at all. They are very appropriately named floating ribs. Now, the bones themselves? They're completely rigid. If they were solidly fused together, locomotion would be impossible. So, our movement relies entirely on joints. While some joints in the skull are fixed, and some in the spine have very limited movement, true locomotion depends on synovial joints. These feature a fluid-filled synovial cavity between the articulating bones, acting basically like biological engine oil. Whether it's the hinge joint in your knee, the ball and socket in your shoulder, or the pivot joint in your neck, these fluid-filled hinges allow the considerable movement required for you to walk, run, and grasp. The crucial point we really have to remember here is that this whole combination of neural signals, sliding muscle filaments, and articulating joints is a highly sensitive, finely tuned system. When this delicate biological machinery breaks down, the effects are profound. Autoimmune attacks like myasthenia gravis or genetic degenerations like muscular dystrophy can severely compromise our muscle control. At the skeletal level, age-related osteoporosis depletes bone mass, increasing fracture risks. And conditions like tetany cause wild, uncontrollable spasms simply due to low calcium levels disrupting those microscopic sarcomere contractions we discussed earlier. We've traveled all the way from the simple shifting of an amoeba to the 206 bones and millions of microscopic myosin heads working together inside of you right now. Knowing that every deliberate, voluntary action you take relies on this incredible, synchronized cellular dance, I want to leave you with this final thought. The next time you get up to walk across the room, feeling the pull of your skeletal muscles and the fluid rotation of your synovial joints, will that simple, everyday act of taking a step ever really feel ordinary to you again? Keep learning, and I'll catch you in the next explainer.